We welcome you, dear students, to the session on varieties of gene expressions. In this session, we will mainly focus on multiple alleles, lethal genes, pleiotropic genes, gene interactions and epistasis. Starting with multiple alleles. The existence of genes is inferred from the processes of segregation and recombination of body characters. Only those genes are known which have an alternative expression. Mendel and his followers used the term allele or allelomorph to denote the alternative form of the normal gene. It means the genes for tall and dwarf characters of pea plant are alleles. The former is normal or wild allele and the later is mutant allele. A gene can mutate several times, producing several alternative expressions. When 3 or more alleles are found for any particular gene, these are called multiple alleles. These occupy the same locus in homologous chromosomes. Multiple alleles are defined as genes that are members of the same gene pair and are located on the same locus. All of them control the same character, but each of the allele affects that character somewhat differently than the others. In any diploid organism, a particular gene pair is represented by 2 alleles of the series, one allele on each of the 2 chromosomes of a homologous pair. This is because an organism has just 2 chromosomes of each type. These form a homologous pair. However, in the individuals of a population or species, many alternative forms of the same gene may be present. The grouping of all the possible alleles of a gene pair is defined as a system of multiple alleles and their mode of inheritance is called multiple allelism. The multiple allele system can be represented as A1, A2, A3, A4 and so on. Characters of multiple alleles. Multiple alleles occupy the same locus within the homologous chromosomes. It means only one member of the series is present in a given chromosome. Since only 2 chromosomes of each type are present in each diploid cell, only 2 genes of the multiple series are found in a cell and also in a given individual. The gametes contain only 1 chromosome of each type, therefore, only 1 allele of the multiple series in each gamete. Crossing over does not occur in the multiple alleles. Multiple alleles control the same character, but each of them is characterized by different manifestations. Sturt Event has summarized it, telling they carry the same function but with varying degree of efficiency. The multiple alleles of a series are more often related as dominant and recessive. More commonly, the normal gene is dominant to all other mutant alleles. Even the intermediate members of the series may be related as dominant and recessive, or they may exhibit codominance. Multiple alleles have been studied in rabbits, guinea pigs, mice, Drosophila and men. We take up the example of wing size in Drosophila. A large number of multiple alleles are known in Drosophila. One of them is the series of wing abnormality ranging in size from no wings to normal wings. The normal wings are dependent on the L plus or VG plus allele. The extreme expression with no wings and just stumps is the result of one allele VG or LVG in homologous condition. A narrow strap-like wing is produced by VGST or LST. Notched wings are associated with the allele VGNO or LNO, and nicked is associated with VGNI or LNI. These phenotypes are produced from the homozygous genotypes, and their heterozygous combinations result in intermediate phenotypes. Wild type, that is normal gene, is dominant over all other members of the series as shown here on the screen. Moving ahead, we will now learn pleiotropic effect of genes. The action of gene at cellular level is unitary, that is, one gene, one action. But some genes produce a broad spectrum of phenotypic changes so that a gene has multiple action. This is called pleiotropy. Lethal genes. Certain genes are known to control the manifestation of some phenotypic trait as well as affect the viability of the organism. Some other genes have no effect on the appearance of the organism but affect the viability alone. These genes are known as lethals or semi-lethals depending upon their influence. Complete lethal genes in homozygous condition kill all or nearly all homozygous individuals, while in case of semi-lethal genes, some homozygous individuals are able to survive. The lethal genes are always recessive for their lethality and express the lethal effect only in homozygous condition. Lethals may be dominant, recessive or conditional. Dominant lethals. The dominant lethal genes are lethal in homozygous condition and produce some defective or abnormal phenotype in heterozygous condition. Their most serious effect in heterozygous may also cause death. A classic example is that of inheritance of sickle cell anemia in man. The disease sickle cell anemia is caused by a gene HBS which is lethal in homozygous condition but has a slight detectable effect in the heterozygous condition, producing sickle cell trait. The homozygous for this gene, that is HBS generally die of fatal anemia. The heterozygotes or carriers for HBS, that is HBA or HBS, show signs of mild anemia as their RBC become sickle shaped in oxygen deficiency. A marriage between two carriers therefore results in carrier and normal offspring in the ratio of 2:1. Recessive lethals. The recessive lethals produce lethal effect only in homozygous condition. Their heterozygotes are normal. Therefore, recessive lethals remain unnoticed in the population but are established in the population. These are detected only when two heterozygous persons get married. The example in support is of Tay-Sachs disease. The recessive lethal gene for Tay-Sachs disease causes death of young children only in homozygotes which are unable to produce enzymes needed for normal fat metabolism. The accumulation of fat in nerve sheaths hampers transmission of nerve impulse, leading to poor muscular control and mental deficiency. Conditional lethals. The genes which may be normal to the individual in a particular environment may prove to be lethal when environment is changed. In Drosophila pseudo-obscura, the flies live normally at a temperature at 16.5 degrees Celsius, but when it is raised to 25 degrees Celsius, the flies begin to die. Penetrance and expressivity of lethals. The lethal genes have been found to differ in their penetrance and expressivity, showing broad range of effects. This may range from cent % lethality, that is no survival, to almost complete viability with no pronounced effect on the phenotype. The maximum extreme may be where it may cause death of the possessor, whether in homozygous or heterozygous condition. Semi-lethals do not cause definite lethality, but decrease the lifespan. It has been estimated that each human carries about 6 lethals on an average. Balanced lethal systems. Lethal genes can be used in establishing balanced lethal system where both homozygous dominant and homozygous recessive die, leaving only the heterozygous phenotype. Such balanced lethal systems are known in Oenothera, Drosophila and mice. Further now, we will study gene interactions. With the rediscovery of Mendel's work, it was thought that Mendel's laws hold good for all situations. Breeding experiments conducted after Mendel and with further advancement of knowledge, it was found that there are several exceptions and extensions to Mendel's laws. Such extensions include multiple alleles, multiple factors, sex linkage, etc. The exceptions to Mendel's laws include incomplete dominance and co-dominance. Pleiotropy, penetrance and expressivity, and the effect of environment on gene expression are other extensions and exceptions of Mendelian ratio. In all such cases, the phenotypic ratio varies, but Mendelian principles of segregation and independent assortment are operative during gamete formation for the distribution of alleles in the gametes. All the cases can be discussed under two categories, that is interallelic or intragenic interaction and non-allelic or intergenic interaction. Interallelic or intragenic interaction. This includes interaction between the genes of an allelic pair. It is further divided into incomplete or partial dominance and co-dominance. Incomplete or partial dominance, that is 1:2:1 ratio. After Mendel, several cases were recorded where F1 hybrids were not related to either of the parents, but the F1 hybrids in heterozygous conditions show an intermediate phenotype. It means that two genes of allelomorphic pair are not related as dominant and recessive. This was discovered by Carl Correns and was described as incomplete or partial dominance. This is also called blending inheritance. In such cases, F1 heterozygotes develop phenotype intermediate between the two homozygous phenotypes. In F2 generation, the phenotype ratio matches the genotype ratio of 1:2:1. Example we glance here is of inheritance of flower color in Mirabilis. In 4 o'clock plant Mirabilis jalapa, when plants with red flowers, that is capital RR, are crossed with plants having white flowers, that is small rr, the F1 hybrid plants bear pink flowers with capital R small r genotype. When these F1 plants with pink flowers are self-pollinated or are crossed among themselves, they produced plants with red, pink and white flowers in the ratio of 1:2:1 in F2 generation. Co-dominance, that is 1:2:1 ratio. In codominance, both the genes of an allelomorphic pair express themselves fully in F1 heterozygotes. It means a heterozygote for codominant gene exhibits both the characters side by side. The coat color in cattle is an example of codominance. When red cattle with genotype R1 are crossed with white cattle bearing genotype R2, the F1 hybrids have roan colored skin and not the intermediate pink. The roan color is actually expressed by a mixture of red and white hairs which develop side by side in the heterozygous F1 hybrid. In F2 generation, red, roan and white appear in the ratio of 1:2:1. The phenotypic ratio coincides with the genotypic ratio as displayed here on the screen. Non-allelic or intergenic interactions. In gene interactions, following observations and assumptions are found. Genes that interact are present on different loci and are unlinked. If complete dominance exists between the alleles of any gene pair or both the gene pairs, such as capital AA and capital A small a, or capital BB and capital B small b, then the same genetic effects are seen. All P1 crosses will involve homozygous individuals, that is capital AABB into small AABB or capital AA small bb into small aa capital BB. Therefore, F1 generation always has only heterozygous genotypes, that is capital A small a, capital B small b. The results of F2 generation are conclusive. With two pairs of genes, F2 genotypic ratio is 9:3:3:1, which equals to 16. That is, capital AABB is equals to 9/16, capital AA small bb is equals to 3/16, small aa capital BB is equals to 3/16 and small AABB is equals to 1/16. In gene interaction, this F2 ratio is variously modified. Collaborator genes is to novel phenotypes, that is 9:3:3:1 ratio. In collaboration, two gene pairs which are present on separate loci but influence the same trait interact to produce some totally new trait or phenotype that neither of the genes by itself can produce. Example is that of inheritance of fruit shape in summer squash, that is 9:6:1 ratio. In cucurbita pepper, the fruit shape is controlled by two pairs of genes. When plants with disc shaped fruits, that is capital AABB, are crossed to plants with long fruits small AABB, F1 fruits are all disc shaped. In F2 progeny, fruits with novel shape, that is sphere shaped, appear along with fruits having parental phenotypes. The F2 generation shows disc shaped, sphere shaped and long fruit bearing plants in the ratio of 9:6:1. The results show collaboration between genes A and B. Lastly, we discuss epistasis. Epistasis, that is act of stopping, is the interaction between non-allelic genes present at separate loci in which one gene pair masks, inhibits or suppresses the expression of other gene pair. The gene that suppresses the expression of other gene is known as inhibiting or epistatic factor, and the gene which is prevented from exhibiting itself is known as hypostatic. Epistasis can be mainly of two types: dominant epistasis and recessive epistasis. Dominant epistasis, that is 12:3:1 or 13:3 ratio. In dominant epistasis, out of two pairs of genes, the single dominant allele, that is gene A of one gene, masks the activity of other allelic pair, that is capital B small b. Since the dominant epistatic gene capital A exerts its epistatic influence by suppressing the expression of gene capital B or small b, it is known as dominant epistasis. Example: dominant epistasis in dogs. In dogs, white coat color appears to be dominant. It develops due to the action of epistatic gene capital I, which prevents the formation of pigment to be produced by hypostatic gene capital B. The hypostatic gene capital B produces black coat, while its hypostatic allele small b produces brown coat color only when gene capital I is recessive. The progeny of dominant gene capital I does not allow them to function and results in white color. When two white dogs are crossed, they produce white, black and brown in the ratio of 12:3:1. Recessive epistasis, that is 9:3:4 ratio. Epistasis due to recessive gene is known as recessive epistasis, that is out of the two pairs of genes, the recessive epistatic gene masks the activity of the dominant gene of the other gene locus. The dominant capital A expresses itself only when the epistatic locus capital C also has the dominant gene. If the epistatic locus has recessive gene small c, gene capital A fails to express. It means both small cc capital AA and small cCAA individuals will have the same phenotype, so that the overall phenotype becomes 9:3:4. The coat colors in rodents, especially in mice, is an standard example. In mice, ancestral coat color is greyish as in wild mice. Agouti coat color is due to the presence of the two pigments in the fur. In agouti color, each hair is black with narrow yellow bands near the tip. Albino coat color is due to absence of pigment and is recessive homozygous. Black coat color is produced due to absence of yellow pigment from agouti pattern. Blacks also breed true. These are recessive to agouti. When black mice are crossed with albinos, the F1 progeny are all agouti. When these F1 agouti interbreed, the F2 progeny has agouti, black and albino in the ratio 9:3:4. Additionally, epistasis can also be distinguished further into duplicate dominant epistasis and duplicate recessive epistasis. Duplicate dominant epistasis or duplicate genes, that is 15:1 ratio. When two pairs of genes determine the same or nearly the same phenotype, so that either of the two genes is able to produce the character, these are said to be duplicate factors or duplicate genes. The recessive phenotype develops only when gene pairs are double recessive. Example is of endosperm color in maize. In maize, the endosperm color is determined by two pairs of genes. When both or either of the two genes are dominant, the endosperm is yellow. The white endosperm develops only when both the genes are recessive. A cross between plants with yellow endosperm, that is capital Y1, Y1, Y2, Y2, and white endosperm, that is small y1, y1, y2, y2, produce plants with yellow endosperm, that is capital Y1, small y1, capital Y2, small y2, and in F2 produce yellow and white in the ratio of 15:1. Duplicate recessive epistasis, also known as complementary genes, that is 9:7 ratio. The complementary genes are two pairs of non-allelic dominant genes present on separate gene loci which interact to produce only one phenotypic trait, but either of them, if present alone, does not produce the phenotypic trait in the absence of other. It means that for the development of the dominant character in question, both the complementary genes must be represented at least by their one dominant allele, say AB. Absence of even one of the two genes produces recessive phenotype, that is small aa capital BB or capital AA small bb. Genes for flower color in sweet pea is evidence of complementary genes. In sweet pea, the purple color of flowers is dominant to white and gives typical 3:1 ratio in F2. White flower varieties breed true and crosses between different white varieties usually produces white flower progeny. But in some crosses between two white varieties produce purple flower plants in F1 and purple and white flowered plants in the ratio of 9:7 in F2 generation. Conclusion: genes are known to control one or more functions and hence are termed as either monotropic or pleiotropic. The existence of gene expression depends on the interaction of genes through segregation and recombination processes. Usually, a gene has two alleles existing one on each of the homologous pair of a chromosome, but some genes have more than one allele affecting the same character differently, and hence the term multiple alleles has been coined. Certain genes show pleiotropic effects and these genes are said to be lethal genes. Gene interactions are also believed to be exceptions and extensions of Mendel's laws. These expressions and interactions commonly include incomplete dominance, complete dominance, codominance, pleiotropism, multiple allelism, penetrance and expressivity, etc. In all these cases, the phenotypic ratio varies with the genotypic ratio, which is well explained with examples in the talk. Thank you.